Chapter 10 Cell Cycle and Cell Division. All organisms, even the largest, start their life form from a single cell. You may wonder how a single cell then goes to form such large organisms. Growth and reproduction are characteristics of cells, indeed of all living organisms. All cells reproduce by dividing into two, each parental cells giving rise to two daughter cells each time they divide. These newly formed daughter cells can themselves grow and divide, giving rise to a new cell population that is formed by the growth and division of a single parental cell and its progeny. In other words, such cycles of growth and division allow a single cell to form a structure consisting of millions of cells. Cell cycle. Cell division is a very important process in all living organisms. During the division of the cell, DNA application and cell growth also takes place. All these processes, that is cell division, DNA application and cell growth, hence have to take place in a coordinated way to ensure correct division and formation of progeny cells containing intact genomes. Haploid number of chromosomes of organisms is called genome. The sequence of events by which a cell duplicates its genome, synthesizes the other constituents of the cell, and eventually divides into two daughter cells is termed as cell cycle. Although cell growth in terms of cytoplasmic increase is a continuous process, DNA synthesis occurs only during one specific stage in the cell cycle. The replicated chromosomes DNA are then distributed to daughter nuclei by a complex series of events during cell division. These events are themselves under genetic control. Phases of cell cycle. A typical eukaryotic cell is illustrated by human cells in culture. These cells divide once in every approximately 24 hours. However, this duration of cell cycle can vary from organisms to organisms and also from cell type to cell type. Yeast, for example, can process through the cell cycle in only about 90 minutes. In bacteria, E. coli, the cell cycle is 20 minutes. The cell cycle is divided into two phases, interphase and mitosis phase. M phase. The M phase represents the phase when the actual cell division of mitosis occurs. The interphase represents the phase between two successive M phases. It is significant to note that in 24 hours, average duration of the cell cycle of the human cell, hence division, will last only for about an hour. The interphase lasts for more than 95% of the duration of the cell cycle. Chromosomes are these condensed during the interphase. The M phase starts with the nuclear division corresponding to the separation of daughter chromosomes, karyokinesis, and usually end with the division of cytoplasm, cytokinesis. The interphase, though called the resting phase, is the time during which the cell is preparing for division by undergoing both cell growth and DNA application in an orderly manner. DNA and central application occurs in interphase. The sequence of cell cycle is G1, S, G2, and N. Interphase. The interphase is divided into three further phases: G1, gap 1, S phase, synthesis phase, and gap G2 phase, gap 2 phase. G1 phase, post-mitotic gap phase. G1 phase corresponds to the interval between mitosis and initiation of DNA application. G1 is the longest phase of interphase. During G1 phase, the cell is metabolically active and continuously grows but does not replicate DNA. But proteins and RNAs are synthesized. DNA polymerase is synthesized in G1 phase. S phase. S or synthesis phase marks the period during which DNA synthesis or replication takes place. During this time, the amount of DNA per cell is double. If the initial amount of DNA is denoted by 2C, then it increases to 4C. However, there is no increase in the chromosome number if the cell had diploid or 2N number of chromosomes. G1. Even after S phase, the number of chromosomes remains the same, that is 2N. In animal cells, during the S phase, DNA application begins in the nucleus and the centriole duplicates in the cytoplasm. Histone protein synthesis occurs during the S phase. DNA polymerase is active during S phase. G2 phase, pre-mitotic gap phase. Synthesis of DNA ends in G2 phase. During G2 phase, proteins are synthesized in preparation for mitosis while cell growth continues. During cell cycle, RNA and protein are synthesized in both G1 and G2 phase. Spindle fiber for proteins are synthesized during G2 phase. Some cells in the adult animals do not appear to exhibit division. Example, heart cells. And many other cells divide only occasionally as needed to replace cells that have been lost during injury of the cell death. These cells do not divide further and exit G1 phase to enter into inactive called quiescent stage, G0 of the cell cycle. G0 stage of the cell denotes exit of the cell from the cell cycle. During G0 stage of the cell cycle, cells divide to undergo differentiation. Cells in G0 phase remain metabolically active but no longer proliferate unless called on to do so depending on the requirement of the organisms. In animals, the mitotic cell division is only seen in the diploid somatic cells. Against this, the plants can show mitotic division in both haploid and diploid cells. N phase of mitosis. Mitosis was discovered by Strasburger. The mitosis was given by Fleming. Cell division is initiated by cytokinin. Cyclin is associated with mitosis. Cyclin is required for the cell's cycle. CDK, other protein essential for completion of cell cycle. Mitosis is divided into two steps, karyokinesis and cytokinesis. Suitable material for study of mitosis is the unnamed root apex or base or nail base. This is the most dramatic period of the cell cycle, involving a major reorganization of virtually all components of the cell. Since the number of chromosomes in the parents and progeny cells is the same, it is also called as equational division. Though for convenience, mitosis has been divided into 4 stages of nuclear division, it is very essential to understand the cell division is a progressive process and very clear-cut lines cannot be drawn between various stages. Mitosis is divided into the following 4 phases: prophase, metaphase, anaphase, and telophase. Prophase. Prophase is the longest phase of the mitosis. Prophase, which is the first stage of mitosis, follows the S and the G2 phase of interphase. In the S and the G2 phase, the new DNA molecules formed are not distinct but intertwined. Prophase is marked by the initiation of condensation of chromosomal material. The chromosomal material becomes untangled during the process of chromatin condensation. The centriole, which had undergone undergone duplication during the S phase of interphase now begins to move towards the opposite pole of the cells. The completion of prophase can thus be marked by the following characteristics of events. Chromosomal material condenses to form compact mitotic chromosomes. Chromosomes are seen to be composed of two chromatids attached together at the centromere. Initiation of the assembly of mitotic spindle and the microtubules and the proteinaceous components of the cell cytoplasm helped in the process. Cells at the end of the prophase, when viewed under the microscope, do not show Golgi complexes, endoplasmic reticulum, nucleolus, and nuclear envelope. Azide and cyanide inhibit prophase, metaphase. The complete disintegration of the nuclear envelope marks the start of the second phase of mitosis. Hence, the chromosomes are spread through the cytoplasm of the cell. By this stage, condensation of chromosomes is completed and they can be observed clearly in the microscope. This then is the stage at which morphology of chromosomes is most easily studied. Chromosomes can be counted best at the stage of metaphase. Metaphase is the best stage to observe shape, size, and number of chromosomes. At this stage, metaphase chromosome is made up of two sister chromatids, which are held together by the centromere. Small disc-shaped structures are at the surface of the centromere are called as kinetochores. These structures serve as a site of attachment of spindle fibers formed by the spindle fibers to the chromosomes that are moved into positional at the center of the cell. Hence, the metaphase is characterized by the chromosomes coming to lie at equator with one chromatid of each chromosome connected by its kinetochore to spindle fiber from one pole and its sister chromatid connected by by its kinetochore spindle fibers from the opposite pole. The plane of alignment of the chromosomes at metaphase is referred to as metaphase plane. The key feature of metaphase are spindle fibers attached to kinetochores of chromosomes. Spindle fibers are made up of proteins, tubulins. Higher plants differ from animals in having anastral spindle. Plant cells lack both centriole and esters. Spindle fibers are of three types: continuous, chromosomal discontinuous, and non-chromosomal discontinuous. Astral rays are a form of microtubules. Chromosomes are moved to spindle equator and get aligned along metaphase plate through spindle fibers at both poles. Colchicine is called mitotic poison because it prevents the formation of spindle since it prevents assembly of microtubules. Colchicine arrests mitosis at metaphase. Mitosis at metaphase. Colchicine is obtained from cones of Colchicum autumnale. Colchicine do not inhibit replication of chromosomes, so colchicine treatment causes doubling of chromosomes and lead to polyploidy. Anaphase. At the onset of anaphase, each of the chromosomes get arranged at the metaphase plate. It splits simultaneously, and the two protochromatids, now referred to as chromosomes of the future or time nuclei, begin their migrations towards the two opposite poles. As each chromosomes move away from the equatorial plate, the centromere of each chromosome is towards the pole, and hence the leading edge with arms of the chromosome trailing behind. Mitosis anaphase differs from the metaphase in possessing same number of chromosomes and half the number of chromatids. Thus, anaphase is characterized by the following key events: centromere splits and chromatids separate. Chromatids move to the opposite pole. Telophase. Telophase. At the beginning of the final stage of the mitosis, that is telophase, the chromosomes have reached their respective poles, decondense, and lose their individuality. The individual chromosomes can no longer be seen, and chromatid materials tend to collect in the mass of the two poles. This is the stage at which shows the following key events: chromosomes clusters at opposite spindle poles, that identity is lost as discrete elements. Nuclear envelope assembles around the chromosome clusters. Nucleolus, Golgi complex, and ER reforms. Cytokinesis. Karyokinesis involves divisions of nucleus while cytokinesis involves division of cytoplasm. Mitosis accomplishes not only the segregation of duplicated chromosomes into daughter nuclei, karyokinesis, but the cell itself is divided into two daughter cells by a separate process called cytokinesis, at the end which cell division is complete. In an animal cell, this is achieved by the appearance of furrow in the plasma membrane. The furrow gradually deepens and ultimately joins the center, dividing the cytoplasm into two. Plant cells, however, are enclosed by a relatively inextensible cell wall, therefore they undergo cytokinesis by a different mechanism. Chromosomes cluster at opposite spindle poles. Their identity is lost as discrete elements. Nuclear envelope assembles around the chromosome clusters. Nucleolus, Golgi complex, and ER reform. Cytokinesis. Karyokinesis involves division of nucleus, while cytokinesis involves the division of cytoplasm. Mitosis accomplishes not only by segregation of duplicated chromosomes into daughter nuclei, karyokinesis, but the cell itself is divided into two daughter cells by a separate process and cytokinesis, at the end of which cell division is complete. In an animal cell, this is achieved by the appearance of a furrow in the plasma membrane. The furrow gradually deepens and ultimately joins the center, dividing the cell into cytoplasm into two. Plant cells, however, are enclosed by a relatively inextensible cell wall, therefore they undergo cytokinesis by a different mechanism. Interphase. In plant cells, wall formation starts in the center of the cell and goes towards to meet the existing lateral walls, center to walls. The formation of new cell wall begins with the formation of a simple precursor called the cell plate that represents the middle lamella between the walls of the adjacent cells. Phlegmoplast is the precursor of cell plate. Phlegmoplast is a part of the spindle which left after chromosomes have moved to the poles. At the time of cytoplasmic division, organelles like mitochondria and plastid get distributed between the two daughter cells. In some organisms, karyokinesis is not followed by cytokinesis, as a result of which multinucleate conditions arise, leading to the formation of syncytium. Example: liquid endosperm in coconut. Significance of mitosis. Mitosis is the equational division is what usually diploid cells only. However, in the lower plants, in the some social insects, haploid cells also divide by mitosis. It is very essential to understand the significance of this division in the life of an organism. Are you aware of some examples where you have studied about the haploid and diploid insects? Mitosis results in the production of diploid daughter cells with identical genetic complements. Usually, in mitosis, daughter cells are genetically similar to parent cell. The growth of multicellular organisms is due to mitosis. Cell growth results in the disturbing the ratio between the nucleus and cytoplasm, and it therefore becomes essential for the cell to divide to restore the nuclear cytoplasmic ratio. A very significant contribution of mitosis is cell repair. Cells of the upper layer of the epidermis, cells of the lining of the gut, and the blood cells are being constantly replaced. Mitotic divisions in the mesenteric tissue, the apical and the lateral cambium results in the continuous growth of a plant throughout the life. Mitosis is within the nucleus is called endomitosis. Meiosis. Term meiosis was given by Farmer and Moore. Meiosis were discovered by Sutton. As compared to mitosis, meiosis is exchange of chromosomal segments. Meiosis can be studied in angiosperms in dividing pollen mother cells, in anther or pollen sac in megaspore mother cells or ovule. Meiosis is best seen in microsporangia. The production of offspring by sexual reproduction includes the fusion of two gametes, which each with a complete haploid set of chromosomes. Gametes are formed from specialized diploid cells. These specialized kinds of cell division that reduces the chromosome number by half results in the production of haploid daughter cells. This kind of division is called meiosis or reductional division. Meiosis ensures the production of haploid phase in the life cycle of sexually reproducing organisms, whereas fertilization restores the diploid phase. We come across meiosis during gametogenesis in plants and animals. This leads to the formation of haploid gametes. The key feature of meiosis are as follows. Meiosis involves two sequential cycles of nuclear and cell division called meiosis 1 and meiosis 2, but only a single cycle of DNA replication. Meiosis in 1 involves two nuclear divisions and one chromosome divides, hence is called a double division. Meiosis 1 is initiated after the parental chromosomes are replicated to produce identical sister chromatids at the S phase. Meiosis involves pairing of homologous chromosomes and recombination between them. 4 haploid cells are formed at the end of meiosis 2. Meiotic events can be grouped in the following phase: meiosis 1, prophase 1, metaphase 1, anaphase 1, telophase 1, and similarly meiosis 2. Meiosis 1. Prophase 1. Prophase of the first meiotic division is typically longer and more complex when compared to prophase of the mitosis. It has been further subdivided into the following 5 phases based on chromosomal behavior, that is leptotene, zygote, pachotene, diplotin, and dikinesis. Leptotene. During leptotene stage, the chromosomes become gradually visible under the light microscope. The compaction of the chromosome continues throughout the leptotene. In leptotene stage, the chromosomes appear thin, long, thread-like. The bead-like thickened portions of the leptotene chromosomes are called as chromomeres. This is followed by the second stage of prophase 1 called zygote. During this phase, chromosomes start pairing together, and this process of the association is called synapses. Such paired chromosomes are called as homologous chromosomes. In synapses, two homologous chromosomes are connected at chromomeres. Electron micrographs of this stage indicate that the chromosome synapsis is accompanied by the formation of complex structures called synaptonemal complex. The complex formed by the pair of homologous chromosomes is called a bivalent or tetrad. However, these are more clearly visible at the next stage. The first two stages of prophase 1 are relatively short-lived and compared to the next stage, that is pachytene. During this stage, bivalent chromosomes now clearly appear as tetrads. Tetrad is made up of two homologous chromosomes, each with two chromatids. This stage is characterized by the appearance of recombination nodules, the sites at which crossing over occurs between non-sister chromatids of the homologous chromosomes. Crossing over occurs in pachytene. Crossing over is an exchange of genetic materials between two homologous chromosomes. Crossing over is only an enzyme-mediated process, and the enzyme involved is called as recombinase. Crossing over leads to the recombination of the genetical material on the two chromosomes. Exchange of paternal and maternal chromosomes material during pachytene is called crossing over. 5 recombination between homologous chromosomes is completed by the end of pachytene, leaving the chromosomes linked at the site of other crossing over. Diplotin. The beginning of diplotin is recognized by the dissolution of the synaptonemal complex and the tendency of the recombined homologous chromosomes of the bivalents to separate from each other except at the site of crossovers. These X-shaped structures are called as chiasmata. In oocytes of some vertebrates, diplotin can last for months or years. During diplotin, chromatids clearly visible in meiotic prophase. Repulsion of homologous chromosomes begins during diplotin. Dikinesis. The final stage of meiotic prophase 1 is dikinesis. This is marked by termination of chiasmata. During this phase, the chromosomes are fully condensed and the meiotic spindle is assembled to prepare the homologous chromosomes for suppression. By the end of dikinesis, the nucleolus disappears and the nuclear envelope also breaks down. Dikinesis represents the transition to metaphase. Metaphase 1. The bivalent chromosomes align on the equatorial plate. The microtubule from the opposite pole of the spindle attached to the pair of homologous chromosomes. A bivalent in metaphase 1 consists of 4 chromatids and 2 centromere. Spindle fibers are made up of protein tubulins. Number of chromatids per chromosome is 2 in both mitotic and meiotic metaphase, but the number of chromatids per chromosome is different in anaphase 2, in meiosis, and in mitosis. If a cell has 2 and 50 chromosomes, then the number of chromosomes grouped at the equatorial plate of metaphase 1 will be 25. Anaphase 1. The homologous chromosomes separate while sister chromatids remain associated at the centromeres. Separation of homologous chromosomes. Anaphase at anaphase is called as disjunction. Segregation of Mendelian factors (TT) occurs during anaphase 1. Polar movement of diodes occur during anaphase 1. In meiosis 1, centromere do not divide. Telophase 1, the nuclear membrane and nucleolus reappears, cytokinesis follow, and this is called as diad of cell. During meiosis 1, the number of chromosomes is halved. Diploid chromosomes number is 8. Then the number of chromatid in each daughter after meiosis is also 8. Although in many cases the chromosomes undergo some dispersion, they do not reach extremity extended state of the interphase nucleus. The stage between the two meiotic divisions is called as interkinesis and is generally short-lived. Meiosis 2. Meiosis 2 is initiated immediately after cytokinesis, usually before the chromosomes have fully elongated. In contrast to meiosis 1, meiosis 2 assembles normal mitosis. Prophase 2. Interkinesis is followed by prophase 2, a much simpler prophase than prophase 1. The nuclear membrane disappears by the end of prophase 2. The chromosomes again become compact. Metaphase 2. At this stage, the chromosomes align at the equator and the microtubules from the opposite pole of the spindle get attached to the kinetochores or the cystochromatid. Anaphase 2. It begins with the simultaneous splitting of centromere of each chromosome, which is holding the cystochromatid together, allowing them to move towards the opposite pole of the cell. Meiosis 2 is the equational division due to separation of chromatids. Telophase 2. Meiosis ends with telophase, in which the two groups of chromosomes once again gets enclosed by nuclear envelope. Cytokinesis follows, resulting in the formation of tetrad cell, that is 4 haploid daughter cells. Significance of meiosis. In meiosis, the daughter cells differs from parent cell as well as amongst themselves due to segregation of independent assortments and crossing over. Meiosis is a mechanism by which conservation of specific chromosomes number of each species achieve across the generation in sexually reproducing organisms, even though the process per second periodically results in the reduction of chromosome number by half. It also increases the genetic variability in the population of organisms from one generation to another to the next. Variations are very important for the process of evolution. Reorganization of genetic materials with the genetic recombination occurs during meiosis. Meiosis is the evolutionary significance because it results in the recombination. Meiosis is the cleavage of nucleus without recognizable chromosomes, distribution and spindle formation. Two reduction divisions and one equational division is called by brachiomeiosis.